Well, good morning, everyone. It's a really great pleasure to be here. I hope I'm a good speaker, too. Gosh, well, it's great to be here, and I can't imagine a better venue for thinking together about this really important moment that we're in with important technologies, not only gene editing, but other technologies that are converging that I think are really giving all of us, and certainly scientists and clinicians and entrepreneurs, opportunities that even a few years ago we couldn't really have imagined being in our hands. It really is a privilege, I have to say, to be part of that. And I thought I would just begin this discussion by pointing out, sort of asking this question, I guess, is how did we get here? And where is here? What is this moment that we're really in? And I thought I would just mention a few things to you to consider that I was thinking about as I was putting together this talk. And so recently I was asked to write a review article for an academic journal on CRISPR. And so I was trying to think about how I would possibly focus, and I was given a 4,000 word limit. So I was trying to think about how would I focus this article, and so I went to PubMed, which is our server that is run for consolidating and capturing all of the research articles that are written on topics, and I just typed CRISPR into the search. And can you imagine, I got 8,400 entries. And if you sort of plot the curve, it sort of looks exponential, right? So the vast majority of these publications have appeared in the last few years, and the real majority have occurred just in the last 6 months. So it's an incredible time. And I was doing this on a weekend, and I was sort of I thought, oh, I'll spend an afternoon doing this. 3 hours in, I said to my husband, "Oh my gosh, I'm going backwards in a time, and I'm only at December of 2017." So it was truly sort of awesome to think about how I was going to try to capture even a fraction of that in the context of a review article. I'm still not quite sure how I'll do that. We're keeping patent attorneys employed, right? So there's also a large, just again, exponential number of patent applications filed around the world by academic organizations, by commercial organizations, and this is protecting all sorts of things, everything from fundamental technologies for gene editing to applications of those technologies, products that are being produced now using gene editing. It's truly, truly awesome, again, to sort of see that kind of exponential growth. And that, of course, is really just a symbol of the excitement around what's happening and the opportunities now for doing things that, again, just in the recent past, none of us really could have imagined being a reality. Here's a few other things to think about. So there are now 3 publicly traded CRISPR companies that have billions in market cap. Think about that. And none of them have products yet, but it's just the anticipation of how the opportunities to use, and these three companies are all focused on human therapeutics. We're not even talking about any of the other opportunities with gene editing. This is strictly in human therapeutics. And there are many, many more startup companies that are not yet publicly traded that are using CRISPR gene editing for everything from agricultural applications to synthetic biology to reagents. Almost sort of any area of biology that you can think of where you might want to manipulate DNA, you can probably these days find a startup company that's trying to get into that space. There are at least 4 clinical trials that I'm aware of that are ongoing in China, in the US, and in Europe. More are for sure going to be underway within the next 12 to 18 months. And, and I think that this is, again, we're sort of at a moment where the progress of those clinical trials will really shape the future, because as long as they go well and people are not harmed by these initial trials, I think that we're going to see a lot more excitement about moving into trials for other kinds of diseases. Right now, these trials are for cancer immunotherapy, so editing immune cells to target cancer, and also for genetic diseases of the eye so far. What else? $250 million have been invested already by nonprofits to support a very specific application of CRISPR gene editing called gene drives, which means using a gene editing technology to introduce a trait into a population and allow it to spread very quickly. Why would you want to do this? Well, there's a lot of interest in this for controlling mosquito-borne diseases. And so these nonprofits that have invested in CRISPR-based gene drives include the Gates Foundation, the Wellcome Trust, and Tata Industries Foundation in India are all investing in this. And there will certainly be more. And this, of course, brings up various concerns about the environmental impacts of this and the ethical and sort of societal implications of doing something that would have potentially a widespread environmental impact, hopefully positive, but people are a little bit, it's a little bit hard to predict how something like that might affect the environment if it were released. Books have been published on CRISPR-based gene editing. There are more books in the pipeline that I'm aware of. Tens of millions of dollars at least have already been spent on one of the very prominent patent fights that I'm unfortunately a part of, and more will be spent, no doubt. And then, of course, there are all of the meetings that are ongoing that are like this one, that are aiming to bring people together to really discuss the ethical and societal implications and scientific implications of this kind of collective moment that we're in. The national academies have already released a report on human germline editing, and there is a second summit meeting that's planned for later this year in Hong Kong to follow up on that discussion. And I know there are efforts going on globally, really, to try to pull people together and initiate discussions about how do we collectively think about regulating something like this. Is it even possible to do that globally? And how do we ensure, or try to ensure, that this technology is deployed in ways that are positive and have a positive impact on human societies? And it truly is, it's sort of one of these mind-blowing things. How did I get here, and why am I standing on the stage? And I thought I would just start by telling you all a little bit about my own involvement in gene editing. And as you heard from Peter's introduction, this is not my original area of research at all. I've been a biochemist for my whole career and a structural biologist. I grew up in science, being fascinated by really very fundamental questions about life and evolution, and I was trained by a great scientist, Jack Szostak, who was fascinated by the role of RNA molecules in early evolution of life. And so I started my career working on RNA molecules that did interesting chemistry and trying to figure out what they looked like, what the shapes of those molecules were, and what that could tell us about the early origin of life. And but really, the story of gene editing, I think, goes to the other nucleic acid, and that's DNA, this beautiful double helical structure that, I think with the discovery of the structure in the 50s, really catalyzed many people to begin thinking about how you could understand this code of life. You could read the sequence. You could eventually, as Peter was saying, technologies that have come along over the last few decades have enabled scientists to make copies of DNA, to sequence DNA, synthesize DNA, clone genes out of DNA. All of these things have been incredibly enabling and have, in each case, I would argue, really pushed science forward because of the ability of scientists to suddenly do things that they couldn't do before and to really control this code of life in different ways. gene editing is sort of the next, I would say, iteration in that progression of technologies. It's a tool that allows rewriting the code of life. And this is sort of the, I had this, this is a slide that I sometimes show when I'm talking to, high school students about this, is, trying to explain that, the idea here is that we can imagine DNA in a cell to be the script. It's sort of the text of a document. And gene editing is the ability to edit that text, not to do that in a random way or in a sort of, hope and pray kind of way, but to do it in a very targeted, precise, accurate, and efficient fashion. And that's really what gene editing enables, is for the scientists to be able to make alterations to the code of life in desired and targeted ways. And the amazing thing about the CRISPR technology for gene editing, and by the way, this is not the only technology for gene editing, but I'll explain shortly why this is a very enabling way of doing gene editing. The crazy thing about CRISPR is that it came kind of out of left field, because it was really developed by a cohort of scientists who were working on something seemingly completely unrelated. And that was really to understand how bacteria fight viral infections. And to sort of give you a little bit of the background to this, the study of bacteria and the viruses that infect them really, again, goes back to, I think, really the origins of modern molecular biology. A lot of the early work that was done to understand fundamental things like the way DNA is copied and the way proteins are synthesized, even the ribosome that, that Venki Ramakrishnan worked on with his colleagues. These are all systems and insights that came from studying bacteria and also sometimes the viruses that infect them. And so, it was sort of widely assumed, I guess, in the field that we mostly knew about these pathways. But, this is the wonderful thing about science and about biology in particular, is that life is complicated, and also life is very, very creative. And if there are ways of doing things that are efficient and effective, you could probably bet that some form of life somewhere has figured out how to do it. And in my own lab, we've been interested for a long time, as I mentioned, in the function of RNA molecules and how RNA can help control the flow of genetic information in cells. So I'm not a microbiologist, and I haven't really done work in my career, much at least, on understanding the way that viral infections affect bacteria. But I wanted to point out that when a virus infects a cell, like this cartoon shows here, this really fundamentally involves the genetic material of the virus being injected into the host cell and starting to take over that host machinery so that more copies of the virus can be synthesized. And so we've sort of, over the years in my own lab, peripherally at least, thought about this process in the context of how RNA molecules might be engaging with viruses and helping the cell to defend itself. And earlier in my career, we worked on this in the context of hepatitis C virus, which is as you know, a virus that infects humans. So I was really intrigued when I learned back in, I think it was around 2006, from a colleague at UC Berkeley, Jill Banfield, about a system in bacteria that is a system that involves cells acquiring pieces of DNA from viruses and then being able to use that genetic material in the form of RNA molecules that can protect cells. And to show you what this looks like, this is a cartoon that illustrates this CRISPR immune system. And by the way, this is really a cartoon put together based on research done in several labs, including a yogurt company and a group in the Netherlands who are doing fundamental microbiology, as well as a group in Canada. So these were all microbiologists that were working either at academic or commercial organizations who were fascinated by this, at the time, really kind of obscure and mostly unknown system that some bacteria had evolved to fight viruses. And the way this was identified originally is that there are, in about 50% of sequenced bacteria, you can find in the genome a highly repetitive DNA element that's called CRISPR. It's an acronym for this. And this element includes a series of identical sequences shown by R, so these are repeats, that flank unique bits of DNA sequence shown in these boxes. And the amazing thing that was published in 2005 by 3 different bioinformatics groups was that these sequences in the CRISPR arrays are often derived from viruses. And so you could find perfect sequence matches between these integrated bits of sequence in the CRISPR arrays and viruses that infect those bacteria. And what emerged over the next few years with some really beautiful bacterial genetics was that bugs that have a CRISPR array can acquire DNA sequence from a viral infection, like this one shown right here, and integrate that new piece of genetic material into the CRISPR array on one end, so it's kind of a growing store of past viral infections. It's almost like a genetic vaccination card, if you will. And then those sequences are copied into molecules of RNA that have these little handles in the structure of the RNA that are binding sites for CRISPR-associated or Cas proteins that can bind to these little handles in the RNA and cut the molecules into functional units that each include one of these integrated sequences in the center, so that part is different in each of these CRISPR RNAs, and then the handles on either end that are identical in every CRISPR RNA. And the cool thing about this is that these RNAs then assemble very specifically with one or more proteins encoded by the genes for CRISPR-associated proteins found typically nearby in the bacterial genome to form complexes of proteins and RNA where the sequence, this unique bit of sequence in the RNA, is the guide. It's the address label that tells this complex where to go. And where it goes is to a sequence in DNA that matches that target site. And when that set of base pairs forms between the CRISPR RNA and the DNA, that recruits the associated proteins to cleave the targeted DNA. So it's a beautiful way that bacteria can use genetic information from an infection to protect themselves from future infection by this sort of surveillance mechanism. So when I heard about this from my colleague Jill Banfield, I was fascinated because, in part, really because of the role of these RNA molecules in providing the addresses for these protein complexes. And I really wanted to understand this, in part because I thought that it was a fascinating evolutionary parallel to a different pathway called RNA interference that occurs in eukaryotic cells, plants and animal cells, even humans, that involves also small RNAs that will allow the cell to target particular nucleic acids, in that case, our other RNAs, and control, therefore, the flow of information coming from the genome. And I thought it would be amazing if bacteria had figured out kind of a different way to use RNA to control the genome like this. And so we started working on this aspect of the pathway, namely how these RNA-guided proteins are able to interact with DNA and achieve cutting. And it was right around 2011 that I went to a conference and I met Emmanuelle Charpentier, who is a scientist who does a very sort of work very distinct from my own. She's a microbiologist and had always been interested in bacteria that are important human pathogens. And when we met at this meeting, she told me about work she was doing on an organism called Streptococcus pyogenes, a human pathogen that causes a flesh-eating disease in immune-compromised patients. And relevant to this microbiology conference, it had a CRISPR system in its genome that had a unique property, at least at the time, which was that it had a single gene, a single CRISPR-associated, that encoded a single CRISPR-associated protein called Cas9 that was shown genetically at the time to be critical for protecting cells against a viral infection. And so when Emmanuel and I talked at this conference, we realized that we had complementary skill sets to figure out the function of this Cas9 protein. And I can tell you that already at that meeting, we were both really excited about this project because we appreciated that this protein was probably very interesting to be an RNA-guided way of targeting DNA in a sort of an addressable fashion. But really testing that hypothesis and figuring out how that might work was the basis of what we worked on together. And that led to a fabulous collaboration that involved postdocs and students in both of our laboratories that led to an understanding of the Cas9 protein, which I'm showing you very schematically here, that this is actually an enzyme, and it's this blue structure is representing the protein, that works by using its CRISPR RNA, this molecule here, to recognize a 20 base pair sequence in DNA and trigger cleavage of the DNA by two separate active sites, catalytic domains in the protein that could cut both strands of the DNA, not randomly, but at one place that was dictated by the position of this RNA base pairing interaction. And a couple of things that emerged from that early study, which was done biochemically using purified components, were critical to thinking in the evolution of our own thinking about this system. And one was that this is an interaction that involves recognition not only of these 20 base pairs in the DNA, but also this short motif in the DNA target sequence that sits right next to the targeted site, and it's called the PAM. And this is critical for the interaction of this protein because it triggers DNA unwinding by a mechanism that we're still fully yet to understand in detail, but clearly is critical for the way that this system operates. And the other thing is that Martin Jinek and Chris Chylinski, the two postdoc here and student who are working on this in our labs, figured out that this reaction requires not just one RNA, but a second RNA called tracer, which was a type of RNA that Emmanuel's lab had originally discovered to be important in producing these CRISPR RNAs in the first place. And these two figured out that the RNA sticks around, it forms a structure on the end of the RNA that is a handle, it's a binding site for the Cas9 protein. So you really need to have both RNAs and this protein to get this reaction to work. And then Martin Jinek, very clever biochemist working on this project in my lab, was trimming away at these RNAs to figure out what the minimal sequences would be for this reaction to work. And that led to realizing that the two RNAs could actually be linked together. So you could make a chemical linkage. This is just extending the RNA sequence, putting in a loop that links the address portion of the RNA here to the handle that binds to Cas9. So this part of the RNA is the same in every case, but this part, the 20 nucleotides on the end, can be altered. And they can be altered trivially, because this is something that we do all the time in our molecular biology labs, is make changes to DNA and RNA sequences like this. And that meant that you could now use this single, we called it the single guide RNA, to address the Cas9 protein to any desired DNA sequence following these rules and selecting a site that had a match to this 20 nucleotide part of the guide RNA. And I think for us, that was really, this is sort of, I can remember this is probably burned into my memory for the rest of my life, is, Martin Jinek doing this experiment to test whether the single guide would work for Cas9, and coming into my office and showing me data that indicated that he could cut any desired sequence in a large DNA molecule by programming Cas9 with different versions of this kind of RNA that just had to change to the sequence right here. And we looked at each other and we really realized that this project had gone from a curiosity-driven research effort to an opportunity to harness this as a technology to do something very different from what it does in bacteria. And the reason for that was that we were working on this at a time when there was a ton of work going on in many labs globally that had led to an understanding of, first of all, how DNA repair works in eukaryotic cells, and also how one could take advantage of DNA repair pathways to trigger changes to genomes in a targeted fashion. And I'm really just going to show you one slide that very simply tries to illustrate this, and that is that unlike in bacteria, where a double-stranded break to DNA typically leads rapidly to degradation, and cells are dividing quickly anyway, so it's hard for them to repair these breaks before something catastrophic happens during DNA replication. But in slower-growing cells like plants and animals, humans, when a double-stranded break happens occurs in the genome like this, the cells have repair pathways to fix the break. And they can do that by either sealing up the gap by sometimes introducing a small disruption to the DNA sequence, maybe just a single base pair could be different, and or on the right-hand side, if there's a template DNA molecule around that has some matching sequence to the flanking ends of the broken DNA, this template can be recombined during the repair pathway to integrate a new piece of genetic information at the site of the break. And so appreciating this, labs for really the last, I would say at least a couple of decades have been trying to figure out how to harness this for gene editing by figuring out how to introduce a double-stranded break to DNA at a desired place. And I can remember back in the 1980s, when I was a graduate student, Peter Dervin's lab at Caltech, very wonderful chemist who had figured out that you could use oligonucleotides that were coupled to chemicals that would cut DNA, could actually be used for that purpose. And in fact, Jim Gusella at Mass General Hospital mapped the position of the Huntington gene, very important for a neurological disorder called Huntington's disease, at that time using that kind of Peter Dervin oligo strategy. And then, of course, in subsequent years, and really, I would say really in the last decade or so, the opportunity for much sort of more sophisticated technologies came along in the sense that these were technologies that relied on programmable proteins that could be engineered in the lab to recognize particular DNA sequences and generate a break. And these were, the most famous of these are called zinc finger nucleases and talons, and these could be highly effective at inducing a double-stranded break at a desired place in a genome. The challenge was that they also required a fair amount of protein engineering, and they were technologies that were not trivial. They were not simple or cheap to use, and so there were in fact companies that were set up to help academics and other commercial groups to prepare the proteins they would need for particular kinds of gene editing experiments, but it was certainly not something you could do in a high throughput way at all, right? It really was you had to be very dedicated, that you really wanted to make a change to a genome at a place and then spend the time to make the engineered proteins that would allow that to happen. And the beautiful thing about CRISPR is that it turns out that all along, bacteria had already come up with a different way of doing this that was much more amenable to sort of high throughput implementation and could be harnessed by scientists because of the very nature of the way the DNA recognition works. Because instead of relying on a protein DNA binding event, which is what these earlier technologies required, with CRISPR, we have an RNA DNA guided interaction that is trivial to change in the laboratory. And that's really the difference with CRISPR. I'm going to show you a video that just briefly illustrates how this, and just sort of summarizes what I told you about these CRISPR systems and how this allowed, understanding how they work, allowed them to be harnessed for gene editing. So this is showing you a virus infecting a bacterial cell. If the cell has one of these CRISPR arrays, then it can acquire a piece of that viral DNA into the array, flanked by the repeat elements, and then the cell is able to make a copy of that entire unit in the form of a CRISPR RNA molecule that is subsequently processed into the functional bits that each include one of these unique virally derived sequences. That combines with a protein, and for the purposes of this discussion, this is the Cas9 protein. Of course, the tracer RNA has to be there. And then these protein RNA complexes, or using the sort of the engineering that I showed you, the single guide form of the RNA, can then be deployed to find matching sequences in DNA, and this can be DNA in the context of an entire genome. When that match occurs, the DNA opens up, the RNA hybridizes with one strand of the DNA, and then the protein cuts the DNA. And when this happens in bacteria, that leads to degradation of the sliced DNA, so it's a great way to defend against viruses. But when this kind of cleavage happens in a eukaryotic cell, as I mentioned, the cells can find that break and repair it by using either the non-homologous end joining mechanism that I described to introduce a small change to DNA, or even by integrating a much larger piece by homology-directed repair. And this was really how the little niche field of studying CRISPR biology led to this sort of watershed moment that we find ourselves in with a really powerful technology for gene editing. And I just want to show you a couple of other things about how this technology actually works. So this is a 3D printed molecular model of Cas9. It's based on an X-ray crystal structure that was solved in Martin Ginek's laboratory at the University of Zurich, where he's now a professor. And what I'm showing you here is the protein, which is the white molecule, its guide RNA, which is the orange molecule, and the way that it interacts with a double-stranded DNA molecule that has a 20 base pair sequence matching the sequence in the RNA. When that match occurs, this protein literally displaces one strand of the DNA double helix to allow an RNA-DNA helix to form inside the protein. And then that binding event activates the two chemical cleavers in Cas9 to make a precise double-stranded break. So you can really predict extremely accurately. We know exactly where the DNA will get cut based on where this guide RNA sits with respect to the DNA sequence by Cas9. And just to show you how we imagine that this bacterial system might function in a eukaryotic cell, to harness this for gene editing, the idea was to put a nuclear localization signal on the protein. That means that allows this bacterial enzyme to get into the nucleus of an animal or plant cell, where it interacts with DNA that's packaged as chromatin, which is what you're seeing here. And somehow this bacterial protein with its guide RNA can deal with that chromatin structure to find a sequence that matches the sequence of the guide RNA, forming that RNA-DNA hybrid and cutting both strands of the DNA very precisely. And then by a mechanism that we don't fully understand yet, it hands off those broken ends of the DNA to repair enzymes that can fix the break, sometimes by integrating a new piece of DNA if it's available in the cell, or also just by sealing up the gap, and in the process sometimes introducing a very tiny change to the DNA sequence. And so when Emmanuel and I published our original work in 2012, what happened next was just amazing. I mean, labs read that paper and quickly started to adopt it for all sorts of applications in different kinds of organisms. And remarkably, this system worked quite well for gene editing in a wide range of organisms. And I think it was even before the end of 2012 that a manuscript had been submitted for publication from Keith Jung's lab, showing that you could actually use this to make modifications in the germline of an organism. That was in zebrafish. You could actually make very targeted changes to the DNA of a fish in the embryo stage that would affect the genetic makeup of that entire fish and its future progeny, right? So it was really clear that this was going to be really a transformational kind of tool. And this is just an attempt to try to show you some of the many organisms that have already been modified using CRISPR-Cas9 based gene editing. And you can see that we're covering everything from plants to insects to human cells to sort of anything that you can envision that has DNA in it can basically be manipulated using this technology. So it really opens the door to all sorts of things. And from my own perspective, I think one of the most powerful things that this has done is to really give researchers a great tool for doing really fundamental discovery-based research in systems that in the past would have been difficult, if not impossible, to manipulate. I mean, I was having lunch with graduate students at the University of Cambridge yesterday, and they were telling me that now for their PhD thesis work, they can create in a week, a knockout, a very specific gene knockout in human cells if they want to do that. In the past, that would have been something that might have taken years to do if you could even figured out how to do it, right? So that's one example of just the sort of the enabling that's happened. I just wanted to mention one in the vein of a vignette about how this technology enables people to do science that they would have never imagined doing in the past. And in my own laboratory, as I mentioned, I'm an RNA biochemist and a structural biologist, but a few years ago, a postdoc named Brett Stahl came to my lab and he said, I want to try to use CRISPR-Cas9 to do editing of genes that affect neurons in the brain, and that would eventually be a technology that could be deployed to treat neurological disorders. And so this is just showing you an example of work that Brett has been doing in our lab with now 2 different mouse models of Huntington's disease that we're working with. Could have never imagined a short while ago that I would be doing animal work in the lab, but here we are. And what Brett has been able to do is to make chemical modifications to the surface of the Cas9 protein that give it cell-penetrating properties. That means that these modified proteins are actually able to get across the neuronal cell membranes and get into the nucleus of those cells and make targeted changes, induce targeted changes to the DNA. And this is showing an experiment that he did where he took those chemically modified RNA-guided proteins, injected them into a mouse brain, and we can do these in a very targeted fashion. So we get modification of just localized neurons in particular areas of the brain. And then this is showing you a slice out of a mouse brain that's received, this brain has received injections on 2 sides here. And this is a mouse in which the cells are labeled such that when edited, they turn red, so we can very easily visually detect cells that have received a targeted gene edit with this kind of strategy. And you can see that you get a pretty decent volume of brain cells that actually get edited. And when you quantify this, we're up to about 30% of the cells within one of these sort of volumes of injected tissue that actually have the desired gene edits. So this is something that is now allowing us to work. We're working now with a team of neurosurgeons at UCSF, my local Bay Area medical school, to see if we can start thinking about moving this into larger animal models and eventually, we hope, into patient trials. We haven't mentioned it specifically yet, but I want to point out that when I think about applications of gene editing and how this is going to impact global human societies, I really think one of the biggest impacts is going to be in agriculture. And this is just one example of an experiment that was done by an academic group, Zach Lipman, who showed that you could use CRISPR-Cas9-based gene editing to make targeted changes to tomato plants that allow these plants to bear 2 to 3 times more fruit than you can get from the starting plant. And why does this matter? Well, if you like to grow tomatoes, that's kind of cool. But I think what this really is a harbinger of is I think the coming wave of using gene editing, not just in the major food crops, but also in a wide range of other kinds of plants that are grown in our backyards or in various niches around the world that can allow us to address things like the effects of climate change and the spread of various kinds of pathogen-borne plant diseases. I think that this is a kind of a really exciting moment, and we're seeing now, at least in the U.S., and it may be true here in Europe as well, I've seen over the last year or two a wave of startup companies in the agricultural space that are all being put together specifically around the opportunities with CRISPR-Cas9 based gene editing. So I think the entrepreneurs are starting to recognize that this is a really exciting time in plant biology and genomics and agriculture. There's also potential to use this in animals, and I just wanted to mention this is actually a slide that came from some work out of the George Church Lab at Harvard, where they have been using CRISPR-Cas9 to make modifications to pig genomes, first to remove endogenous viruses from these genomes, and then more recently to actually try to humanize these animal genomes in ways that will allow them to potentially be better organ donors for human patients. And this is sort of another kind of facet of how CRISPR-Cas9 based gene editing is likely to impact human health in the future, not just by directly leading potentially to clinical treatments, but also to allowing the use of animals and plants that have been modified in ways that will be beneficial. And finally, I just wanted to mention germline editing. There's been a fair amount of discussion about this, and I know we'll discuss it later today. This is a slide that shows what we mean by germline editing. So this is you're seeing a pipette here that's holding on to a fertilized egg. This is a mouse egg, fertilized egg, and there's a needle coming in, injecting the CRISPR-Cas9 editing molecules into this early embryo. And when that kind of editing happens at a very early stage in development like this, then those DNA, those genetic changes become part of the entire organism, and they can also be passed on to future generations of that organism. So this is a technology that was used very early on. I think maybe Rudolf Janisch at the Whitehead Institute was maybe the first to publish using germline editing in mice with CRISPR-Cas9, but it's now been adopted by many labs and doing this kind of editing in plants and animals. The question is, should we do this in humans, and will we do this in humans? I would argue we will, and I think it's a question of how it's done and how that might be regulated. I just want to show you an example of this and what this kind of germline editing looks like in a very visual example. I got these slides, the next two slides I'll show you, from a student working at MD Anderson Cancer Center in Texas. And she told me about experiments she was doing in frog embryos. And the slides were so beautiful, I asked her for the pictures. This is the work of Sunata Kadka, a student there. And what you're seeing here are these two-cell embryos, frog embryos, in which the student was able to inject CRISPR-Cas9 into one of the two cells in such a way that she knocked out a gene encoding a brown pigment for the frog and replaced it with a gene that encodes a green fluorescent protein. So you can see you get very precisely one of the two cells is fluorescent green, the other cell is not. And this editing is precise. So the DNA is not negatively impacted in the sense that these edited embryos can go on to develop into animals. And when you look at the tadpoles that develop, here's what you see. So on the top, you can see an animal under regular light, where if you look carefully right down the middle, you can see that the top cells on the top half are making this brown pigment protein, and the cells on the bottom half are not. And if we put that animal under a fluorescent light, you can see that the cells on the bottom are all green. So they've all got that genetic change. This kind of editing has, of course, been done in other animals, as I mentioned. And I think for me, it was really this paper that was published in early 2014 using CRISPR-Cas9 in monkey embryos that really, sort of made me realize that I needed to be getting involved in a public discussion about the potential for this to also be used in human beings. And this has led to a series of meetings. A number of people here in the audience have been part of that. The Royal Society has been very intimately involved in those discussions. And it's also led to a lot of sort of public attention to CRISPR-Cas9. And this, those of you that read The Economist might remember this picture from, it's already maybe a couple of years ago, that was on the cover of The Economist under the banner Editing Humanity. And it was really all about the potential to use human germline editing to make changes to human DNA that would be heritable and would not only lead to potentially curing genetic disease, but also to what we might call enhancements. And I should point out that most of these things right now we don't really have any way of doing because we either don't know the genes involved or we know that there are many, many, many genes involved, so it would be technically difficult, if not impossible, to do today. But I think that this really is a very important topic to be discussing because we're clearly on the verge, I would say, of having the technical capability to do at least some kinds of human embryo editing, potentially in a clinically relevant fashion, and we have to grapple with what that really means. The National Academies put out a report on this topic, and you know, this was published in February of last year. And it's clearly a moving target because since then there have been two papers published very prominently in the journal Nature and then a number of others that have appeared elsewhere that have shown that you can do CRISPR-Cas9 based gene editing in viable human embryos. And there are debates about you know, sort of the details of the interpretation of those data, but there's no doubt that this is something that is moving forward quite quickly in various countries. I just want to mention that, you know, one of the books I alluded to earlier is one that I wrote with a graduate student in my lab. So Sam Sternberg was a student who did work was sort of working alongside Martin Jinek in the laboratory at the time that this early Cas9 research was done in our lab. And so when he finished his PhD, he told me that he really wanted to write a book about this experience of being in the lab. He sort of recognized that this was a very special, kind of unique time in science. And so I really give him the lion's share of the credit for taking a year out of his professional life and spending a year doing the research and then writing the first draft of this book that we ended up putting together about our experience. And then finally, I just want to mention, you know, so a number of people have asked me, how do you grapple with you know, the challenges of this technology? And you know, I run an academic research lab. I teach undergraduate students at Berkeley, and you know, I have a son at home, so I wear a lot of hats. And so I asked myself the same question: how do I handle this? And I decided that one thing I could do would be to involve a lot of other colleagues. And so with support from both UC Berkeley and UCSF, we were able to start the Innovative Genomics Institute back in 2014. And this is an academic organization that works with AstraZeneca and a number of other companies, but we also do a lot of work with our academic colleagues to both do fundamental research. It's mostly on very sort of specific applications of CRISPR-Cas9 based gene editing for sickle cell disease, for example. But we also have a team that focuses on the ethics and outreach that we think needs to be done. And I encourage you to visit our website and to engage with us. And I think there are going to be important ways that this organization can actually work together with the Royal Society and other groups that are trying to disseminate good, you know, high-quality scientific information in an understandable fashion so that we can really invite a global conversation about this really exciting moment that we're in biology. Thank you.